Chapter 7, Part 4, Metabolic Pathways. So this is where the fun begins in this chapter. Um, so this is something that you should have touched on in A&P 1. Some classes go a little deeper than others, but we're gonna try to keep it as simple as possible because this is kind of a confusing topic. So when we talk about catabolism, there are things that happen in a very kind of complex multi-step series. They refer to them as pathways. So with each step in each of the pathways, there's something that's catalyzed or broken down by an enzyme. So the product of one reaction comes out of that reaction and moves on to the next step of the pathway and is then catalyzed again and then broken down, and then that's broken down and goes into the next step. So many pathways have branches that provide alternate methods for nutrient processing. And I will say this, this is something that it never kind of was brought to my attention, so I didn't really pay it. Like I didn't get it. I didn't think about it. But we talk a lot about like aerobic respiration, and um, and there's other types of pathways like aerobic respiration. We're talking about the breaking down of glucose in order to extract the energy from that, the ATP essentially, or what we end up with is ATP. But there's other pathways that break down other things like lipids and proteins and such. So, it was never kind of brought to my attention over the years of learning all this stuff, and it wasn't an obvious thing to me. And maybe that's a downfall for me, but the fact is that there's other things that are metabolized in order to extract energy. The one we always talk about is like aerobic respiration or fermentation, and glucose. Okay, so know that there's others. We're not going to talk about them. I think the book touches on them, but I kind of skip over those in this. So these pathways are not individual, standalone. They are interconnected and they merge at many sites. So, so we're going to talk about, and this should look familiar. You should recognize the Krebs cycle. You should recognize glycolysis. I'm not going to worry really about this, but what you should know is when the Krebs cycle happens in glycolysis, there's enzymes that are used in there to break down certain things. Now, you're not going to have to know all the enzymes, but being aware that that's part of the deal. That's part of these reactions. We need these enzymes and these cofactors and these coenzymes in order for these things to happen and for products to be kind of tossed out of these either cycles or linear reactions. And this is just showing that there are linear reactions like glycolysis. There's cyclical reactions, which is an example would be the Krebs cycle, which is also known as the citric acid cycle. And then there's branching systems. I'm not going to ask you about the linear versus cyclical versus branched. I'm going to focus on specific types of pathways, such as glycolysis and aerobic respiration and fermentation. All right, so most energy we know is stored as ATP. Energy is managed in the form of chemical reactions that involve the making of bonds or breaking of bonds, and the transfer of the electrons in the process. So an exergonic reaction releases energy as this reaction goes forward. Exer releases an X, you release your X, right? Exergonic releases energy, and endergonic require energy in order for them to happen. So these often are coupled so that released energy is immediately put to use. So as energy is released, it is then moves on to an endergonic reaction. Cells possess specialized enzyme systems that trap the energy present in the bonds of nutrients as they are progressively broken. So energy released during exergonic reactions stored in high energy phosphate bonds in ATP, and ATP temporarily stores and releases the energy in these chemical bonds. All right, so redox reactions, so oxidation and reduction. So oxidation is the loss of an electron, and I want to point out that an electron is charged negative. It's got a negative charge. So when we talk about the loss of negatives, then you have to think of that as becoming more positive. So if we take away negatives, it becomes more positive. Reduction is the gain of electrons. And again, if electrons are negatively charged, then you're becoming more negative. Okay? It's kind of opposite of what you would think, at least in my brain it is. So when a compound loses electrons or becomes more positive, it's oxidized. And when a compound gains electrons, it's reduced or becomes more negative. Okay, so oxidation reduction or redox reactions are common in the cell and are indispensable to the required energy transformations. All right, so I'm going to keep this simple. Redox reactions always happen in pair, or always kind of associated as pairs. So as one agent gives up an electron, it immediately goes and is gained by another agent. So sodium here gives up an electron on the outer shell. I'm not going to get into too much chemistry here, but immediately as this gives away an electron, it becomes more positive. It becomes positive because these are neutral initially, right? These are neutral. These are perfect little neutral. Sodium and chlorine is what this one is. And then when the sodium gives up an electron, it becomes positive because we lost some negative. And the chlorine then becomes chloride and is reduced in the process and becomes negative. So as we lose an electron, we become positive, and when something gains an electron, it becomes negative or more negative, depending on if it was already negative, but we're not going to get into that. But we're usually talking about this initially as they're neutral. You lose an electron, the sodium becomes a positive ion now, and the chlorine becomes chloride ion. Because remember, an ion is a element that has gained or lost an electron. All right, so when we talk about, I'm going to skip over some of this, so don't get too excited. It's there for your reference if it comes up on the Smart Book or the homework, but I try to keep this really simple. So a redox pair is an electron donor and an electron acceptor involved in a redox reaction. So it's basically, both parties in the reaction. And by the way, so we're going to talk more about NADH and FADH later, but where they come from is their coenzyme carriers are nicotinamide adenine dinucleotide, mouthful, and flavonoadenine dinucleotide. So NAD and FAD are both neutral. So these are carriers that are going to be involved in reactions later. So we're just introducing you to these guys right now. So I don't care about this, so you can skip right over that. So oxidation reduction reactions salvage electrons and energy that they contain. So I want to put a little footnote in all of this, in that whenever we talk about electrons floating around, going from one molecule to another, or from one, from one uh, participant to another, then you need to remember that there's always going to be a hydrogen involved there. There's always going to be a hydrogen attached to that electron. And that's important knowledge for later on down the line when we get into the actual Krebs cycle and the electron transport chain. So oxidation reactions salvage electrons and the energy they contain. Energy now present in the electron acceptor can be captured to phosphorylate or add a phosphate to. When we phosphorylate something, we add a phosphate. When we Mrs. Rhodes something, we add my stamp to it, right? When I put my mark on it. So energy now present in the electron acceptor can be captured to phosphorylate or add a phosphate to ADP or some other compound to store energy. The cell does not handle electrons as separate entities, but rather as part of an atom, such as hydrogen, which contain the proton and electron. So that's what I meant when I said there's going to be a hydrogen attached or participating in this movement of the electron. So, all right. So a little bit on ADP. I'm sorry, adenosine triphosphate, ATP. So it's a 3-part molecule. It has adenine, ribose, and then 3 phosphates. Okay, we know this from anatomy, hopefully. I'm not going to really ask you a whole lot more about that other than what makes up ATP, adenine, ribose, and 3 phosphates. There's a picture of it. Again, you're not going to have to draw this, but I like to keep this picture in here because you can see the adenine, you can see the ribose, and then you can see these 3 phosphates. And you can see where this phosphate is connected and this phosphate is connected. There's the possibility of energy when they're broken, or the release of energy when they're broken. So ATP utilization and replenishment make up an ongoing cycle. So ATP is broken down to ADP and then later built back up to ATP. So it activates individual subunits before they're enzymatically linked together. They're used to prepare molecules for catabolism, and when ATP is utilized, the terminal phosphate is removed or broken off to release energy and ADP. So ATP is adenosine triphosphate. So that means there's 3 phosphates. ADP is adenosine diphosphate, meaning there's 2 phosphates. So if I have 3 phosphates and break one off, I now have 2 phosphates. So adenosine triphosphate, all 3 phosphates are there. I break one of the phosphates off, and now I have adenosine diphosphate and some energy. So in order to put ADP back together with that phosphate, in order to bind these 2 back together, we need some energy. So if we take some energy, add it to ADP and that phosphate, and now we have ATP again. So it's this back and forth. It's broken down, it's put back together. It's broken down, it's put back together. So basically that energy that is either released or needed to put it back together is just traded back and forth in chemical reactions. In heterotrophs, the catabolic pathways provide the energy infusion that generates the high energy phosphate to form ATP from ADP. And I know this is confusing, so just bear with me here. We're just kind of putting the rug down underneath the whole process that's happening later on down the line. So I'm going to stop here, and then I'll pick up on more of this in part 5.